I'm interested in the relationship between neural activity and behavior. And I think that a way to try and understand this is through recording and manipulating neural activity as an animal is performing a behavioral task. So what I want to do today is really describe an approach in systems neuroscience and try and give you a sense of what is experimentally possible for us right now. And so I'll do this by talking about work I did during my PhD in the lab of Karl Swoboda. And the starting point for this approach, it's really a behavior where you, the experimenter, can not only record and monitor the relevant sensory inputs and motor outputs of the animal, but also control and manipulate them. So as you sort of try and design a behavior, though, I think it's important to think about the animal that you're studying. And we use mice as they have a number of features that make them amenable to study, for example, like genetic model organisms. And if you think about most rodents in general, they live underground, and they're sort of nocturnal animals, and they use their whiskers to navigate through these tunnels, find food. This animal, you can just see he sort of ran through to find some seeds. And you think this is, okay, this is a very, very natural behavior. The animal very cool, but how could we possibly study this in the lab? Because as he runs through this environment, you have no idea what he's actually touching, where he's going. You can't control the environment. And as he moves his head, it's very hard to sort of make high-quality recordings from his brain. And so what we wanted to do was kind of translate that sort of behavior into a lab setting where we can make more controlled experiments. And so here, there is a head-fixed mouse that is running on a trackball, and there are walls located on either side of the animal that are moved with motors in a manner coupled to his locomotion. And so using this system, we can simulate winding corridors for him to run in, really tactile environments for him to explore. We can reward him with water, in this case, for making the correct choices. And what's nice is that we can then record a lot of the important sensory variables. For here, I'm showing a zoom in of his whiskers. This is slowed down by maybe a factor of 10. You can see the individual whiskers touching the objects, bending as they touch the walls. And we can really get a sense of what are the inputs to the animal. And so if you look, what does this behavior actually look like? Here I've unfolded the mouse's trajectory from the ball into the 2D plane. You can see him run left and right in this sort of randomly winding corridor. But you could also put him in more sort of regular structured environments, give him mazes, give him choice points, ask if he goes left or right. And really, what is the animal doing here? Well, he has to sort of sense how far away he is from the walls and then guide his behavior appropriately. And while this, again, may seem like a very simple thing for the animal to do, we actually really don't understand how it is that he would do this, and really even where in the brain this would be happening. And so I think the first thing that you want to do when you have a behavior like this is to start doing silencing of neural activity to try and find the areas of the brain that are relevant for the task. So if we look at a sort of top-down view of the surface of the mouse brain, you can parcel it out into sort of different areas, sort of motor areas, sensory areas for the different senses, sort of somatosensation, the whiskers, vision. And then you want to selectively silence these areas. And what we can do now is we can make a transparent skull preparation on the animal. And then we can do this in animals which have inside their brain a light-activated silencer, so that when we shine light with a laser onto a different part of his brain, we can silence the neural activity, we can shut off the neural activity in that part of the brain. We can then monitor the effect on his behavior and thereby try and figure out, okay, what parts of the brain are involved in this. And so by moving the spot around to different areas, we can then sort of survey the whole brain. And just to give you an example of what this looks like, here you have an animal that's sort of normally making turns, going left and right. then we silence the sort of the whisker part of his brain, just sort of transiently during this turn period. You can see he can no longer make these turns. And so this sort of a technology allows you to identify, okay, where do you want to start looking? But really, once you have a region, you then want to go in and you want to start recording activity as the animal is doing this task. And so in this particular example, the animal is running along, the wall comes in, and then the animal turns and moves away from it. So he's sort of measuring how far he is. And we can now electrically record the activity of neurons. And so what this plot at the bottom shows, each dot represents an action potential or a spike of the neuron. These spikes, this is really the language that neurons use to talk to each other. And here, each line represents a trial. There's time along the x-axis. You can see these are very temporally precise measurements. And this particular neuron, this becomes active when the wall comes in, is only sort of transiently active. Other neurons that we can sort of simultaneously record, this example shows more sustained activity. This neuron actually gets transiently inhibited. The number of spikes goes down as the wall comes in. And this neuron gets inhibited and remains off. And so by making these sorts of recordings as the animal is doing the task, we can then make correlations between the sensory inputs, the walls, the neural activity, and the behavior of the animal. Now, this is sort of nice. With the electrical recordings, you get very good temporal information about what's happening, but you're sampling from a much more limited number of neurons, maybe 30, 100 simultaneously, depending on your technology. Whereas if you want to sort of ask what's going on a much larger spatial scale, we can then use optical methods to record neural activity. And so what I'm showing you here is we've removed part of the skull of the mouse and replaced it with a glass window in this cranial window preparation so that we can then look in with a microscope. And now, remember, right at the beginning in this behavior, we head-fixed the animal so that we can now put this microscope above him as he runs around. And we can do this in animals where we have put inside a genetically encoded indicator of neural activity, a fluorescent indicator of activity. And if we zoom in on a little small field of view there, and you can now see on the right-hand side individual neurons. Those look like these round blobs, these donuts, which are labeled with a fluorescent calcium indicator. Calcium is a proxy for the activity of the neurons. And if I show you now a movie of what this looks like, you can see these neurons, this is highly sped up, I should say, blinking on and off. And you then want to ask, well, how does the blinking on and off of these neurons relate to the behavior of the animal? And so what we have been doing is a pixel-wise regression analysis of the time series of each pixel with a behavioral variable of interest, in this case, where the wall is, using cluster computing, in particular Spark. And why this is necessary is these data sets, they're very large. You have activity of many, many pixels, a million pixels simultaneously recorded for hours at a time. And here, what I'm showing is each pixel has been colored according to the results of that regression analysis, the peak wall distance that makes that pixel most active. The brightness has been has been mapped to the R squared of that regression, and so only the pixels with a reliable tuning are showing up as colored. I sort of then zoom in on a couple of individual examples, individual regions of interest, and then make tuning curves to this wall, the distance the animal was from the wall. You can see the top example that was colored blue is tuned to very close distances. The middle example, that is the lighter blue, is tuned to sort of more intermediate distances. And then this orange example, this actually gets inhibited by further distances. And so the imaging methods are nice because you can assess the activity of many, many more neurons simultaneously. But what you then want to do is follow this up. Let's say you have a coding hypothesis. You take the first neuron, for example. It's the closer the wall, the more active it is. want to follow up that sort of an idea with a perturbation experiment, an activation experiment. What we can then do is also using optical methods, but now instead of using optical methods to record activity, we can use them to drive activity. And what we find here is that by activating the right part of the whisker area of the mouse's brain, we can bias the animal into moving away from the wall, maybe fooling him into thinking he's touching it stronger than he actually is. And we can go one step further now, and here's a mouse like before running around, but we have entirely removed the actual walls that were there. And now, just by activating his brain in the right way, we can fool him into thinking the walls are actually there and get him to make a left turn. You'll see here he's running straight, and then we'll start stimulating him in the right way, and he'll go to the left as if he was sort of running in this corridor, but now we're really just directly activating his brain. And so this sort of says, okay, we have some maybe understanding of how this system is working. But where we're sort of going next, I mean, this is still a very crude manipulation, I think, is the sort of world of online analysis for performing functionally targeted experiments. So what do I mean by that? Like a map like this, you then really, instead of sort of activating all the neurons, you want to do experiments such as activate only the blue ones or only the orange ones. So if you use the results of your analysis to inform how you make your experiments. And so what I've been doing with Jeremy is performing online analysis using the Spark streaming system. So what happens here is we are acquiring these images at the rig as the animal's performing the task. Then we are shipping them off to our local cluster where we are performing analyses. Then we're shipping the data back to the experimenter at the rig in the form of these maps, which now you can watch evolve, and which can then be used for guiding experiments. So either specific activation of neurons, targeted physiology recordings from functionally identified neurons, or maybe things like you have a model for how you think the behavior is being executed that you're going to learn from the data, and then you're going to perform more of like a brain-machine interface style experiment where you use the results of that model that you previously acquired to feed back into how you're driving the sensory stimulus in real time with the animal. And those sorts of things, this sort of model-specific testing, I think it's really key to sort of validating hypotheses and something which clearly requires a large data infrastructure, both for handling that data, shipping it around back and forth, and then deciding what analysis metrics or analyses to perform. So, I really just wanted to give you just an overview of what's possible right now in systems neuroscience. I think these sort of technologies, it's a very exciting time to be there. I'm happy to talk more sort of tomorrow, I think, with people maybe about what are the sorts of questions that we're now going to use these technologies to address in neuroscience. I think that's, I think we can do a lot right now, but then deciding what are the right questions, the right experiments to do, I think that's an enormous part of how we're going to make progress. So with that, I just want to thank a few people, particularly my supervisor, Karl Svoboda, without whom none of this would be possible. Yuri Vlasov, a visitor to the lab, who made the electrical recordings that I showed, and then Jeremy Freeman, who has been instrumental in getting the Spark analysis, both in the offline and online modes, working. So thank you very much.